In recent years, we have seen something quite unexpected. Young people, people under the age of 50, have started getting colorectal cancer. And until now, we haven't really seen the reason why are they getting colorectal cancer. Now, in this recent study, we were actually able to look at the cancers of these young individuals and to compare them to the cancers of older individuals. And we saw a striking difference. This striking difference was mutations generated by colibactin, which is a mutagen generated by a bacteria in the gut. Our study indicates that this bacteria has been present very early in life, probably within the first 10 years of life. It has generated many mutations at that time, and subsequently increased the risk of these children to develop cancer when they became young adults. So what we have known before is that colibactin is found in colorectal cancers. But what people have not looked at is the early onset colorectal cancers. So in overall colorectal cancer, or colorectal cancer in older individuals, you'll see colibactin in about, 10 to 15% in patients. Now, when we look at these younger individuals, we see that at more than 50% of patients. Not only that, but we see that some of the earliest driver mutations, the ones that actually cause colorectal cancer, are being generated by colibactin. Yeah, well, the notion of driver mutations has been around for a long time. In principle, these are the mutations that allow the cancer to progress and to develop. And the important part is who generates these driver mutations. In this specific case, we see very strong evidence of colibactin generating this particular driver mutations. If you look at lung cancer of smokers, you see a very strong evidence for tobacco smoking generating the driver mutations in these cancers. I think prior research, well, first of all, we know that early onset is a huge problem, and prior research hasn't been really able to show something very different for early onset colorectal cancer. This research, for the first time, shows that colibactin is one of the very big differences between early and late onset colorectal cancer. But it should be very clear, it's not only E. coli that can generate this colibactin. There is a number of other bacteria that can also carry the so-called PKS island that generates colibactin. So there could be other infections. E. coli just happens to be the most well-described one. So early onset colorectal cancer is very rapidly increasing in the US. In fact, the US was the first country to report that. But at the moment, we're seeing over 20 countries around the world reporting exactly the same increase. So we're seeing a global problem, something happening on a global scale. And initially, when we started doing this specific study, we wanted to understand these global patterns. We wanted to understand why some people around the world get more cancer compared to other people who get less cancer. And that essentially led to this study design, getting patients from different parts around the world and trying to understand both the country-specific exposures as well as the global changes that can lead to colorectal cancer. So what this study shows very clearly is that colibactin is a mutagen. It generates many mutations, including the ones that are relevant for cancer development, and it does it in early life. Now, this is an associative evidence, which means that further work will be required to show unequivocal causation. The problem with this further work is that it may take many decades. The ultimate proof is an interception or a prevention study where you actually prevent the exposure and show that people do not get cancer. Now, in this specific case, if you're talking about intervening in children, first of all, we don't know how to intervene, but even if we imagine how to intervene in these children, we'll need to wait 40, 50 years to see whether they do not develop cancer, which is a rather long study. Now, in the meantime, one can generate in the short term, the next 2 to 3 years, one can generate additional associative evidence that further implies that there is a causation. But again, the ultimate proof takes decades. So there is at least 2 sets of questions one can ask based on this study. Has one been exposed as a child to colibactin? And has that impacted their risk? And the second question is, what can we do to prevent this exposure? Now, we're working actively to answer both those questions, but the reality is we do not know the answers at the moment. However, this study gives us the foundation to further investigate it. For the first question, we can use these colibactin mutations as a screening tool, and we're trying to develop a stool sample collection protocol that allows us to screen for colibactin and to be able to actually identify young people who are at high risk for developing early onset colorectal cancer. The second part, which is a more complicated part, is saying, well, we know children are getting these infections. We know that they're getting mutations. We are uncertain why that's happening. And there is a number of reasonable hypotheses one can put. These are hypotheses of the mode of birth. These are hypotheses whether children have this breastfed, what kind of food have they taken as children, whether they have taken antibiotics or not. Each one of those is completely reasonable. Each one of those needs to be investigated to understand what causes it. And again, one needs to think also what prevents it. And there is reasonable ways one can use probiotics to do that, but that will require extensive further research. And again, we're talking of a time frame, 5 to 10 years, to be able to fully understand it and to be able to have a solution to prevent this infection in children. At this very moment, I don't think there is anything, as a parent of 2 young children, I don't think there is anything that I can do or a parent can do to check whether their child has had such an infection or has it, because it's asymptomatic. The thing that's going to happen is you're going to get mutations, the child is going to get mutations in their colon, and then subsequently have something happen 30 years down the road. So we do not have the apparatus to monitor this at the moment, at least at the clinical setting to the best of my knowledge. Now, we can develop this and we should develop this, but that will require further research and development. So there are 2 next steps potentially. One of them is, hey, can we monitor people who have the infection to make sure that we capture the one at higher risk? And the second one is, can we prevent people from, or children in this case, of getting the infection? So in this specific case, the next step is further research and development and moving a lot of the things we have developed now to actual practical technologies and products so they can impact the lives of patients and the lives of children.